Hello everyone, welcome to the class, and today we are going to study a very interesting and important chapter of your science notebook, science NCRT notebook. That is this chapter's microorganisms, friend and foe. You might be looking at these creatures in the image here that looks like alien creatures. So you might be wondering what are these and what are these creatures. You will get all your answers. You will get your all questions answered in this session. But before starting the session, I would like to develop some curiosity in you about the chapter. Have you ever wondered how how the curd is made from the milk? You might have seen your mother preparing curd from the milk, and you might have seen your mother taking an inoculum of curd, means a small part of curd, and then she will mix it in a bowl of milk and then keep it aside overnight. And the next day, what you get? You get your curd ready. So, how curd is getting prepared from milk? Have you ever wondered about this phenomena, about this process? In this chapter, we will learn the science behind curd preparation. I'm very sure most of you like bread. You like recipes made from bread, like bread sandwiches, bread rolls, etc. Have you wondered how this bread is so fluffy, so soft to eat, so yum to eat? Pizzas and sometimes your cake, pastries, breads, these are so soft. Have you ever wondered how are these so soft and fluffy? So we will also learn the science behind bread making in this chapter. You might have observed around your surroundings that whoever living being take birth have to die someday. This is a law of nature, whether it is plant, whether it is animal, whether it is we as humans. So when these things die, their body get decomposed naturally in the environment and it get mixed with the soil. So how microorganisms contribute in this process, we will learn the science behind decomposition in this chapter. I'm sure near any river or pond you might have seen such green color substance. What is this substance? Doesn't come in the category of plant, neither it is a kind of aquatic life. What is this? We will learn about this in chapter. We will learn about algae in this chapter. Also, in the rainy season when we don't eat bread for a long period of time, so if it becomes rancid. It develop over it some green and grayish color structures. So what are these structures? And then this bread obviously is unsuitable for consuming. So why how things get spoiled, how microorganisms spoil the things. We will also learn about this in chapter. We know we human beings get ill at certain times and then doctor prescribes us medicine and sometimes the disease is infectious, sometimes it's non-infectious. You have already learned about disease, that there are two kinds of disease, communicable and non-communicable disease. You have already done in your previous classes. So in this chapter, we will learn about various airborne, waterborne and vector-borne diseases, and some microorganisms cause the disease while some organisms protect us or help us to recover from the disease. So we will learn about that in this chapter also. I'm sure you have heard the term antibiotics or have seen antibiotic or have even taken antibiotic at time. So doctor prescribes antibiotics and then we eat it to get recovered from the illness sometimes. So we will learn the science behind antibiotic in this chapter, what antibiotics are made up of, how some microorganisms are beneficial because antibiotics actually are composed of microorganisms, certain microorganisms. So we will learn about that in this chapter. Then virus and vaccines, these are not new terms to us. In the recent times, the world has suffered from coronavirus, pandemic it was, and then we took the vaccine to prevent such virus. So we will learn the science behind viruses and vaccines, and this is going to be really interesting for you. Now, food preservation. So let me give you an example. There was a girl named Pinky, and she bought some mangoes, some raw mangoes, and not raw mangoes means those yellow and sweet mangoes she bought from the market. And then due to some circumstances, she was not able to eat those mangoes for some time. And after a week when she observed the mangoes, they were unsuitable for consuming because they developed over them those dark color patches, means they were not fresh anymore. So then her mind pondered about the fact that when her mother prepared the mango pickle from the raw mangoes, and she store it for a longer period of time, it gets even more tastier, it gets even more tangy. So then she got confused that how it is happening. So we will learn about food preservation techniques in this chapter, why we store food in airtight containers. You might have observed the chips packet and various other food items that you purchase from the market that they come in the airtight container. So why we preserve the food in such manner, how we preserve the food, we will learn more about this in this chapter. So this was quick intro about the various facts and various processes that we are going to learn about in this chapter. And the list don't end here actually. This was just a kind of short trailer, brief trailer for you that what you are going to learn is in this chapter, but the whole picture is still remaining. Yes, and let's start the chapter now. Now let's understand the meaning of the heading of your chapter, that is microorganisms, friend and foe. The word microorganisms. First, let's break it into two parts, micro and organisms. Now, what are organisms? This is a very simple term that you might have heard in your real life, daily life. And organisms actually mean living beings. You know, this means what? It means living beings. And we humans, plants, animals, we all are organisms. So when someone asks about us, what are living beings? Generally, we think about humans, plants, trees, birds, fish, lion, various wild animals, domestic animals, etc. But we generally don't include microorganisms. But the science says microorganisms are also living beings. Why they are living beings? Because they actually show all the characteristics, many of the characteristics that a living being possesses. Now, what is the meaning of the word micro? Micro means tiny, yes, very small, very minute. Even such minute that we can't see them with the naked eye, means without any using any instrument, without having a specific instrument, we can't see them because they are so tiny. They are so tiny. So we need a specific instrument for viewing them. We will learn about that in the further slide. Now let's move to the next part of the heading. What is it? Friend and foe. Friend you all know. You all have friends, so I don't need to explain the meaning of friend. And what is foe? Foe means opposite of friend, means those persons, those things which are not useful for us, not beneficial for us. Generally, we call them foe. So, in this chapter, we are going to learn about that how microorganisms behave as friend to us and how at times they are not friend to us, means they are opposite of friend to us, means they are harmful for us. So we will learn about this in this chapter. So foe versus friend, how microorganisms are useful to us and how certain microorganisms are harmful to us. That is very important to learn in this chapter. You can look at the images here. Foe. So what is the meaning of foe? I am writing here so that you always remember. If you don't know, enemy, and friend. Friend, I am not going to write the meaning of this. You already know. You can sense the meaning from these pictures also. Here is the definition of microorganisms. Microorganisms are microscopic organisms that cannot be seen with the naked eye. What is the meaning of naked eye? Means unaided eye. We need a specific instrument to observe to make them visible. So what we use? We use a microscope. So we use a microscope to zoom into them and then closely observe them. So we can also add a point here that microorganisms are those microscopic, because they are microscopic organisms, they can only be seen under a specific instrument like a microscope. And generally, these organisms are unicellular in nature. Now, what is the meaning of unicellular? It means they are single cell. Unlike we humans and other complex organisms like many plants, animals, so they are multicellular. You will learn about this more in the cell chapter. We human beings have evolved over time, and other organisms like us have evolved over time, and we are not primitive. We are not that simple. Our body has organ system. Organs are made up of tissues are made up of many cells, and also it's not just one kind of cell in our whole body, but there are multiple types of cell in our whole body. But these are microscopic organisms. These microscopic organisms are primitive in nature, and many of them are generally found to be unicellular, means they are just a single cell. And one more important thing is here that you will notice the mushroom image here, and you will say it is not microscopic. We can easily observe it. We can easily see it. Even we can hold it in our hand. So it is a category. It comes in the category of fungi, and it is a macroscopic species of fungi, macroscopic example of fungi. So we will discuss it later in the other slide, but let's just take it like that most of the microorganisms are generally found to be unicellular in nature. Now, I would also like to mention here the specific sizes 10 to the power minus 3 meter to 10 to the power minus 6 meter. So this is their range, size range. Okay, and if you don't understand, I will also write it. You can also write it like it is 1 by 1000 power of 1, and means this is small. You can imagine there is one part and they are so small, means we are dividing it by here 1 2 3 4 5 6. How many zeros? 6 zeros. So this is the range of their size. I hope this is clear to you. Then we will move to the next slide. There's a microscope. So this instrument I was talking about, microscope, you might have seen this instrument in your science lab in your school, and there we put a small sample of pond water. You can take pond water, drops of pond water, or you can take that rancid bread that we discussed in the previous slide that has been raised patches that is not suitable to eat, and that bread you can keep it in the slide that is there. We keep it in a slide, and then we observe it closely. We zoom it, and then we are able to observe the microorganisms like this. In this slide, there is a small sample, and then when we look closely, then we find microorganisms moving around like this. Okay, so here also written that microorganisms can range in size from 1 micrometer to 1 millimeter, and microorganisms can be seen with the help of a microscope. Now here is a list of subtopics that we will cover in this chapter. Microorganisms, this part we have already discussed in the last slide, and this one, where do microorganisms live, we will study further, and all other topics like microorganisms and us, what kind of relation we have with microorganisms, and how they are beneficial to us, mainly we will learn about this. Then we will see harmful microorganisms, and we will learn about the food preservation techniques, and very important nitrogen fixation concept and nitrogen cycle. So we will cover all these subtopics in this interesting chapter. Now the question is, where do microorganisms live? Where these tiny creatures live? They are cosmopolitan. What is the meaning of cosmopolitan? They are everywhere. They are found everywhere. They are in the air, they are in the water, they are in the soil. As a matter of fact, they can be on the surface of our hand. That's why the parents and elder keep telling you, wash your hands before eating anything so that germs don't enter your body. Now, what are germs? Are germs different than microorganisms? So germs also come in the category of microorganisms. But germs are generally considered to be notorious, means they are harmful for us, but they are also microscopic because you can't see germs with the naked eye. And okay, so I hope this is clear. And one more thing that I was telling you in this slide, that where they are found. So they can be found in the pond ecosystem. They are in the ocean, they are at the extreme cold climate weather conditions, and they can even survive in the hot springs. They can even survive in the desert, means they can be there at in the extreme climatic conditions. They can be there in the normal conditions, like they are found in the soil. Some microorganisms grow on other organisms, and some exist freely. Okay, they are even inside our body also. They are in our digestive tract, and some, as I told you, that microorganisms can be friend, they can be foe. So those microorganisms which are naturally in our body, like in our digestive tract, they help to digest food properly. They help in the digestion. And sometimes the microorganisms enter our body like invaders, and they make us ill. So they can be friend and foe. Now, after knowing about where these microorganisms are found, let's study their classification, because classification is very important. When we study by classifying, we study better, as you know. So classification, when we classify microorganisms, we broadly classify them in 4 major categories, that is bacteria, protozoa, fungi, and algae. We will study we will study them in detail in the upcoming slides. Now you can also see here virus. Obviously virus is also microscopic, but we don't generally put it under the umbrella term of microorganisms. Why? Because when we say organisms, as we studied in previous slide, that organisms are living beings. Now virus, virus are kind of slightly different. Okay, that's why we don't group them as bacteria, protozoa, fungi, and algae, because virus can only survive in the body of host organism. To survive, to show the characteristics of a living being, they need a host. They need a host cell. So outside the body, they are just inert. They are there. They are present there, but like they can be in the air. They are present there, but they won't be called living. be living, they will be showing the characteristics of living after they enter into a host cell, after they enter into a host body. What they do then, they take the charge of the cell, they take over the cell, and then they start to replicate themselves, they start to make more and more of themselves, and that's how the virus survives. So because virus is inert when it is outside the body, outside the host cell, so that's why we can observe it like virus is dependent on the host cell to be called living. That's why we don't generally put it under the umbrella term of microorganism. And one more thing, one important thing that I would like to tell here is that when we study, like if we are studying about the animals, then animals are not just one kind. Like we study the water animals are there are then aquatic animals, okay, they are water aquatic animals, then land animals are there, and then there are the animals that fly in the sky. So there are broad variety that we see in the living organism. So similarly, there are various varieties of plants also. Similarly, when we study microorganisms, they are so diverse, so much diversity is there when we study them. That's why we have broadly classified them in the 4 categories to study them properly. Now, first of all, talking about bacteria. Bacteria are very primitive organisms. Primitive what? I mean by primitive means they are very simple. But they come in various sizes, various shapes. Like you can see in the image, this is spirulum. This is a spiral shaped bacteria. Then there is bacillus, and then coccus and vibrio. Coccus are somewhat sphere shaped, while bacillus are rod shaped. Although it doesn't look like rod here, but it is actually rod shaped. This is spiral shape. That's why, as the name suggests, is spirulum. And here vibrio, as you can see, they are comma shaped. So 4 major types I have mentioned here. And bacteria are unicellular, again single cell, prokaryotic. Prokaryotic, you will study about this in detail in the cell chapter. Prokaryotic, if I tell you in brief, means they are primitive. Prokaryotic organisms are those organisms whose nucleus is not well defined, means their genetic material is not enclosed in a nuclear membrane, their genetic material is not properly enclosed. So they lack they lack the well organized structure. So they are prokaryotic organisms. We human beings are not prokaryotic organisms, we are eukaryotic, we are advanced. Okay, we have the cell membrane, our cells have the cell membrane, our cells have the nucleus membrane, genetic material is properly enclosed in our cell. So we will learn about, I won't go into detail of it, we will study that in the cell chapter. And second point says some bacteria are useful for humans while some can be harmful. That we have already studied in the starting of the chapter, that microorganisms can be harmful for us, can be beneficial to us. Similarly, bacteria, obviously they come in the category of microorganisms, so they can be harmful or they can be useful to us. One important thing that you need to remember for the exam is the example. Yes, they can ask you examples also. So they can ask, like, give some example of bacteria. Then you should remember some names, some example of bacteria. So here I'm writing down. You can also note down. Okay, let's use some different pen for writing the examples of bacteria. There are bacteria again is a very diverse group. Okay, here we find various kinds of bacteria, but broadly, mainly you can remember 2 names that I'm writing here. First is rhizobium. Because I am telling this name because this you will learn about more in the nitrogen cycle. Rhizobium bacteria, and one name you can remember is Lactobacillus species. Okay, so these 2 names are important. Curd preparation. So how curd is prepared? There is let's see, there is a milk in a bowl. Okay, and we then add some 2 to 3 teaspoons of curd into it. Inoculum, small part that contains microbe, small part of a substance that contains microbes is known as inoculum. So 2 to 3 spoons of curd we will take here. Okay, and we will keep it overnight. And next day we will find that the curd has been made. Okay, so how this is happening? Milk has some lactose, okay, in it. What milk has? Milk has lactose. Now what happened is that 2 to 3 spoons curd that we have taken here, it contains what? It contains this lactobacillus bacteria. What it contains? Lactobacillus bacteria. So Bacterium lactobacillus भी बोल सकते हैं इसे. We can also call it Bacterium lactobacillus or lactobacillus bacteria. It is there. Now this lactobacillus species will act on the lactose present in the milk. Okay. So here I am writing it as in the short lactobacillus bacteria. Okay. So this will act on this lactose that is present in the milk. And then what it will make? It will make lactic acid. It will act on the lactose present in the milk, and then what it will make? It will make the lactic acid. And that's why you see that the curd has thicker consistency than the milk. It is not flowy like milk. It has slightly stiff texture. Now actually what is happening here, this lactic acid will act on the proteins present in the milk, and a lot of chemical changes will take place there, and then that's why you see the difference in the taste also, difference in the texture also, and your curd is made. So what is game changer here? Game changer here is the lactobacillus bacteria. So lactobacillus bacteria promotes the formation of curd. That this point is very important that you have to remember, because it multiplies in milk and it converts it into curd. And bacteria are also involved in the making of cheese. Not only curd, but in the preparation of cheese and pickle and other food items also bacteria, various kinds of bacteria are used. Now sometimes students get confused with the yogurt also. Yogurt, which is an example of probiotics. You might have heard about this term probiotic yogurt, which actually looks like curd, sometimes also slightly it tastes like curd also. So that's why I have also mentioned probiotic here. And curd itself is a kind of probiotic. Actually, probiotics are live bacteria and yeast. They are those microorganisms which are useful for our digestive system, which actually helps to improve our health. As the name suggests, probiotic means four. Pro means four, and biotic means life. What means pro? What is the meaning of pro? Pro means four, and biotic means life. So they are for improving our life. When we take the probiotics regularly in the appropriate amount, what happens is they help in improving our digestion. And even there are certain microorganisms which are already present, naturally present in our body, and they are there in our digestive tract, and they help in the digestion process. Like our bowel movement, if we take them in appropriate amount, they can help in the bowel movement also, for getting rid of the wastage in our body. They can aid digestion. They can improve your appetite also. So these are probiotics. Okay. And curd preparation that I have already mentioned, that I have already told, it is also important. So this was the useful aspect of bacteria. But we also learned that microorganisms are not always beneficial for us. Sometimes they are harmful. So this is not just about the bacteria, but this is about the microorganisms. That those microorganisms which can cause infectious disease in their host, they are known as pathogen. Like I tell you, in the above picture, in the previous slide, we saw there is this girl who got sour throat, and her throat really hurts. So she's getting sour throat. So some bacteria invaded from the ear into her through her mouth, and then they hurt her throat. So here bacteria are acting as foe. They are acting as what? They are acting as foe here. And here a bacterial infection is really hurting person, causing the disturbed ease, that is disease. And now to get to improve the situation, to get relief in such situation, this girl will do, she will go to doctor, and now the doctor will do what? Doctor will prescribe her some antibiotics. So antibiotics are those organic compounds that inhibits and kill microorganisms, and usually they are used to target bacterial infections. Antibiotics are used to treat what? Bacterial infections. Generally, they are used to target bacteria. Antibiotic, now this is also a very important question, that how antibiotics got discovered. Who discovered antibiotics? So here you can see the name Alexander Fleming. In 1929, he accidentally discovered penicillin. Penicillin is a kind of fungi that inhibits the growth of bacteria. So he was actually researching on something else, but suddenly he got this accidental discovery that he was doing research on some bacteria, and then he observed that the growth of a certain kind of fungi was actually inhibiting the growth of the bacteria there. Then he from that discovery, he discovered the penicillin, and penicillin is a kind of antibiotic that helps to treat the bacterial infection, because in this case the penicillin will do what? It will inhibit the growth of bacteria, and when it will weaken the bacteria in this way, then obviously over a period of time, after completing the course of antibiotic, what will happen? Our body will recover from the bacterial infection. Now, antibiotics, there is very important point to be that we should keep in mind when we talk about the antibiotics. That is the precaution. Antibiotics is not something that you self-prescribe to yourself. When you will go to the doctor will give you a prescription, and he will strictly ask you to take medicines as he suggests only. Okay, he will say that, okay, take this medicine, this kind of penicillin or a kind of antibiotic, one in the morning, one in the evening, okay, and take it in this manner for 5 days. Okay, he say, for example. But some just assume that you feel good in 2 to 3 days only, and then you will stop taking medicine. Then what will happen? Then actually these kind of bacteria are very smart. They will develop resistance. Next time when the same attack will be happening to that person, then at that time, if you will take the antibiotic, it will not be able to treat you. It will not be able to cure you, because the bacteria that invaded your body will develop resistance if you will not complete the course. And sometimes what happens, some more smart people, some more wise people do what? They eat the medicine for more days. Like the doctor said that take this medicine for 5 days, and these smart persons will do what? They will eat the medicine for 7 days. That we will take some extra medicines to make sure that we never fall ill again. But this is actually also very harmful for the body, because there are certain kind of useful microbes that are present in our body. Now, what will happen? Actually, uh, these kind of antibiotics are not that smart that they will be able to recognize the difference between the harmful and the useful bacteria in such a case. And they will then what do? They will do they will kill the harmful. They will kill the useful microbes in our body. They will start killing the useful microbes in our body, and that will be also harmful for our body. So it is very important that you take the medicines only for the prescribed time, only in the prescribed manner. So this is also important. Antibiotics. Keep in mind that these are organic compounds, and they kill the microorganisms or inhibit the growth of microorganisms. Here, when we are discussing the bacterial diseases, then let's just discuss some bacterial disease. So when we study the disease, what we mean by disease, we have seen this in the previous slide. Disease means disturbed ease. So when disease is caused by bacteria, so the mode of transmission can be air, mode of transmission can be water, mode of transmission can be food. So some bacterial disease are seen in humans, some are seen in animals, some are seen in plants also. What is important here is to remember the name of these diseases, because this is in your NCERT notebook. So remember the name of disease that I am going to write here is very important here. So if I talk about the humans, in humans we see many bacterial disease, like there is cholera, there is typhoid, and there is tuberculosis, TB. So these three diseases are seen in humans. Okay, cholera is spread through water, contaminated water. Typhoid can be spread through contaminated food and water, while tuberculosis can be spread through air also. Okay, mostly it is spread through air. While in animals also we see some bacterial diseases are also seen in animals, and very common is the anthrax. This table has been given in the NCERT notebook, so you can refer to that table also. So here I am just giving you brief detail about that. Anthrax. Anthrax. In such a disease, what happens is the cattle generally suffer from anthrax, and they suffer from high fever, they develop blisters on their skin, and this can lead to even death in the animals. And when I say about plants, so bacterial disease can be seen in plants also. So the very famous bacterial disease that is seen in plant is citrus canker. Citrus canker. You can remember this name. And in this disease, what happens is the leaves develop dark patches, and the growth of the plant get affected in this kind of disease, citrus canker. And one very interesting fact that I want to tell you, sometimes students get confused in the cyanobacteria, blue-green algae. So they get confused whether it is algae or whether it is a bacteria. because we call it cyanobacteria is also known as blue-green algae. Actually, when it was discovered, I want to give answer to this also, this confusion point also, because here the things can get slightly tricky. So when that was discovered, actually at that time scientists thought that it's a kind of algae, but later they discovered that the better fit for categorizing the cyanobacteria blue-green algae will be bacteria category. Though then they put it into the bacteria category. So we also know it as now as cyanobacteria. But initially it was called the blue-green algae. So remember this point also. And we will learn more about the cyanobacteria when we will learn about the nitrogen fixation, when we will learn about the nitrogen cycle, then we will get into detail of it. Okay, so let's move to the next slide till then. Okay, fungi. Okay, fungi is very important. So fungi is also a very important microorganisms that we need to know about in this chapter. Fungi, as we had already discussed in its previous slide, that when the bread get rancid, it developed over it green and grayish patches. What are these called? These are called bread mold. Okay, so it is also a kind of fungi, and the yeast that we will further discuss in the upcoming slides is also a very important fungi example. And then there is mushroom. So fungi is actually a weird type of microorganism because it is so much diverse. Okay, there are some fungi that are microscopic, and some examples of fungi are there which are macroscopic, like the mushroom, because mushroom you can easily see it. This is not microscopic. You know the difference between microscopic and macroscopic. Micro means very small, macro means something that we can see, that is large enough to see with the eyes, naked eyes. So mushroom, we can hold it in our hand, we can easily see it. So this is the example of macroscopic fungi. So but it comes in the category of fungi. Fungi, when we talk about, we have already learned about this in our previous chapter, that fungi are actually, they take nutrients from the dead and decaying matter. They are saprotrophs. We have learned about saprotrophs in our previous chapter, okay, in our previous class. So saprotrophs are fungi, means fungi are saprotrophic in nature. In nature, they are saprotrophic. They obtain their nutrients from the dead and decaying matter. Now, there are fungi which are microscopic in size, microscopic, like yeast, commercial yeast that we use for baking purposes, okay. And then there is this fungi that you see on the surface of the bread. You can see it like something is there, but you can't, for observing it closely, you have to use microscope. Then the mushroom can easily seen with the eyes. So let's discuss about the fermentation process, because yeast, which is a very important example of fungi, that is used in the process of fermentation, and fermentation itself is a very important process. Fermentation is very important process. In fermentation, sugars get converted into alcohol. So if fermentation definition is asked to you, then what you will say, fermentation is a process in which sugar get broken down or get converted. So conversion of sugar into alcohol. Okay, fermentation. Now, how this happens? Fermentation happens due to the presence of yeast, because all this conversion is happening due to the yeast. And what is yeast? Yeast is a kind of fungi, and fungi is a kind of microorganism. So microorganism is contributing in the process of fermentation. That's why this process become important. And what are the application? So before discussing the application, let's see the process of baking of a bread, how bread is made. So let's suppose we are going to make a bread, and then we mix the flour which is used to make the bread, and we add a little sugar into it, because sugar is important. And although some sugars are naturally present in the flour, and but let's add some sugar, and then make a dough of it, and some water also, and then let's put some yeast also in this mixture. Yeast is commercially available, you all know. So when we do this, and then after making such a dough, we put a piece of cotton cloth over it, and keep it aside for some time. And after some time, what we notice that the dough has risen to a certain level, means the volume of dough has been increased. So volume of dough, like this was our bowl, and then we see that the dough has been rising in the volume. So that is the reason why these breads and the cake cake breads, etc., pastries, etc., are so fluffy. You see, they are so fluffy, they are so soft because of the rising. And now how this rising is happening, I will also tell you that. So when the when the yeast acts on the sugar that are naturally present in the flour, or we have added some sugar in the dough. So when this yeast act on the sugar, then what happened? What is released? What is made here? Alcohol. Plus in this process, carbon dioxide is released. So actually, and also energy is also released here in this conversion process. Yeast multiplies themselves, and the more and more they multiply themselves, the more the carbon dioxide will try to escape the dough, and the more the carbon dioxide will try to escape the dough, the more the rising of the dough will take place, and the dough you see that becomes so fluffy in nature. So yeast reproduces rapidly, and what it produces? It produces carbon dioxide. In the one more important thing is that I missed out the point, that we are putting a cloth over the dough so that less amount of oxygen is available in this process, because it is very important. Fermentation process. In the fermentation process, the conversion should be done anaerobically. What is the meaning of anaerobically word? Means in the less amount of oxygen. Means oxygen shouldn't be there, or it should be minimal. That's why we are putting this cloth over the dough to make this process happen anaerobically. Bubbles of gas fill the dough. What are the bubbles? Which bubbles of which gas? The gas here is the carbon dioxide. Bubbles of carbon dioxide gas fill the dough, and that's why the volume of dough we see here. It increases the volume. So this is the basis of use of yeast in the baking industry. So in the breads, pastry making, cake making, again we saw a important application of microorganisms, important use of microorganisms in the food industry. In the previous slide we saw lactobacillus bacteria helpful in the formation of curd. In this slide we saw the helpfulness of yeast in the fermentation process, and there are other kinds of fermentation also. This was just one kind of fermentation. There is acetic acid fermentation, there is alcoholic fermentation. Alcohol is prepared from barley, grapes, pineapple. In all these products, sugars are naturally present, and yeast, when added, it utilizes the sugar, and then alcohol is produced, and carbon dioxide is also produced in the fermentation process. So this was all about the fermentation process, and one more important thing is there. Uh, who discovered this fermentation process? The name you should remember, Louis Pasteur. He discovered this fermentation in 1857. Okay, let's move to next slide. But before moving to algae, let's discuss some fungal disease also that I forgot to tell you about. Fungal diseases. So remember some fungal diseases. Like in humans, you can remember there is a fungal disease known as ringworm. I will tell you only those which are important from the exam point of view here. I won't go in much detail, but ringworm is a fungal disease that you can remember, and also in plants that is seen. A fungal disease is seen in plants also, that is rust of wheat. So they can be, the mode of transmission here can be direct contact. Due to direct contact, these disease can spread, and in plant, it can spread through air and seed. Now, talking about algae is another important microorganism category. And here, you need to just remember some example of algae, like Spirogyra, Clydomonas. Remember, these two examples are very important: Clydomonas and Spirogyra. And also algae. If I give you a real life example that I already told, near the leaves and the ponds, you see these green color sticky substance, that is a kind of algae. And algae and fungi, when live in a mutual relationship, that is known as symbiotic relationship. We have already done in the previous chapter, where algae give the food to the fungi, and fungi give the space to live to the algae, and they live in a symbiotic relationship. And algae is another important microorganism. And okay, there are no certain important disease or anything else that you need to learn about here. So let's move to the next slide. That is about the protozoa. Protozoa is also important microorganism. Some examples of protozoa are amoeba and plasmodium. And paramecium is also another important example of protozoa. And one important disease in which protozoa microorganism is a causative, means where protozoa is responsible for the disease, is the malaria. So malaria is caused by microorganism, that is Plasmodium. This Plasmodium causes malaria, and Plasmodium is example of protozoa. So these remember these examples: amoeba, Plasmodium, paramecium, and we will. It is also unicellular. Like amoeba is a unicellular microorganism. Mostly are unicellular here. We will learn more about these in other chapters. And malaria is a disease that is seen in humans. So one important point there out is in the algae, that algae contains a chlorophyll. So if you will see, you will take a sample of this green color substance, sticky substance, and you will put in the slide of the microscope, and you will then find that this actually looks like this. You will be able to observe it. So algae contains a chlorophyll, that's why it is green in color. Now let's move to vectors and carrier. What are vector and carrier? Vector is an organism that can transmit a disease from a healthy person, so from an infected person to a new person without being infected itself. What is it saying here? That suppose this is a healthy person here. So this is a healthy person. Okay, this is a healthy person, and there is this infected person who has been infected by some microorganism, some disease. Okay, and this is a healthy person, and this is, let's say, this is a sick person. Now, what will happen? Suppose a mosquito, whether it is female Anopheles mosquito or Aedes mosquito, come buzzing here, and then it bite this person. Then what will happen? The microorganism or the pathogen, we call the microorganism that causes the disease pathogen. So pathogen will get stick into, or pathogen will get transferred into the what, to this mosquito. And then what will happen? That when this mosquito will again buzz and go to this healthy person, and this mosquito will transmit that pathogen in the it will suck the blood, and then it will transfer that pathogen in the body of this healthy person. So in this whole scenario, what is happening here is that this mosquito, which it is just acting as a carrier, and more technical term is a vector, because it is carrying the pathogen from the body of a sick person. It suck the blood, the whatever the pathogen is here, it get transmitted to this carrier or vector, and then it goes and then go to the healthy person. Then here there also it suck the blood, and then it transfer the pathogen there. But in whole process, this carrier vector is not getting affected. Means जो mosquito यहाँ पे है, it is not getting affected. इसको कोई disease नहीं हो रही है. It is completely fine. इसको कुछ नहीं होगा, but that healthy person will get affected. So here is this Aedes mosquito, Anopheles mosquito is there, and so vectors are only carriers, because what they are doing, they are transferring the disease. Even housefly is also example of vector. Housefly that is commonly seen. So what it do? It will sit on the uncovered food, and also it also sit on the unhygienic places, where various unhygienic places it will sit on, and then it will also sit on the food, and those uh, pathogens will stick in the legs of the housefly, and then when it will sit on the uncovered food, and it will spoil the food also, because that food will then get those pathogens will get transferred into the food, and when some healthy person will eat it, so that can lead to food poisoning, that can make that person sick. So these are the carrier or the vector. These vector or carrier are just acting as a transmitting agent here. And one thing that you also need to remember here is that what agents are Anopheles and Aedes. Means they spread what kind of disease. So you should remember that Aedes mosquito, this is Aedes mosquito, and it causes dengue or dengue. Okay, and this is Anopheles mosquito, and it causes malaria. Means malaria, it transmits the malaria protozoa Plasmodium. Okay, malaria causing protozoa Plasmodium, and Aedes mosquito is agent for dengue disease. Important part and interesting part of the chapter, that is virus. Virus are not new terms to us. Virus we all know, and we have learned about this in the previous slide also, that how virus is different from other categories of microorganisms, that is bacteria, fungi, etc. Viruses are organisms that possess nucleic acid but lack replicating machinery. Means they have the replicating machinery only inside the body of the host cell, that we have already discussed, and it cannot survive without a living cell, and that's why it is sometimes considered to be as a it is not exactly living being, because it is considered to be on the borderline between the living being and non-living being. Means living and non-living entities. entities. It is somewhat in between them, in the borderline, at the borderline. And there are some example of viruses like there is a rabies virus. Some disease are there which are caused with the virus, caused due to virus. Now, virus is very dangerous, and so far we don't know any virus that is useful. Only virus cause disease, and there are certain disease that are seen in the humans, like hepatitis is there, which is caused by virus, and rabies is there, coronavirus is there, measles disease is caused due to virus, and chickenpox is also another example of viral disease, which is in which the mode of transmission is a direct contact. With person develop the blisters over the skin, and when another person come in the contact of those blisters, it get transferred, it get transmitted. The virus get transmitted, and another example are the polio, hepatitis, as we already know. And mode of transmission can be air, can be water, can be the direct contact. And viral disease are not only fatal in humans, but it can affect the plants and animals also. Some important viral disease means some important viral disease that we need to remember in case of plant and animal are, one is already written here, that is mosaic virus. Mosaic virus, a yellow wing mosaic of bhindi, that is mentioned in your textbook also. So, yellow wing mosaic of bhindi, this is a disease which is seen in plant, and the mode of transmission here is insects. And if it asks the viral disease in animal, then remember one name here, that is foot and mouth disease. Foot and mouth disease. It is seen in animals and cattle where they develop blisters over the skin, and fever, high fever is there. It can be fatal for them. So these were the viral disease. And one important thing that you need to remember in the case of viral disease, and there are certain other disease also in which the antibiotics are not effective. So as we already learned about this in the previous slide, that antibiotics are effective only in the case of the bacterial disease. But in case of the viral disease, antibiotics are not effective. Why they are not effective in the case of viral disease? Because mechanism in which the antibiotics work is not effective in the case of viral disease, because sometimes viral disease happen and the symptoms are shown in the body after many days. Like there are certain virus disease when the symptoms are not shown directly, and the best defense system in the case of viral disease is that is our immune system. Our own body's immune system is important, plays an important role here. If our body's immunity is good, so that virus will not be able to survive in our body. But if our own immunity system is low, and then it can be fatal for our body. And one more important thing there here is the antibodies are also important. Antibodies, and don't get confused in antibodies and antibiotics. Antibiotics, what are antibiotics? Antibiotics are the chemical substances or the medicines that we take to kill the bacterial infection in our body, to get cure of the bacterial infections in our body. But antibodies are somewhat these are kind of proteins, and especially these are kind of Y-shaped proteins that are produced by our body in response to the pathogen that enters into our body. Sometimes when foreign particle or invaders or pathogen, harmful microorganism, when they enter into our body's own immune system, the army of good cell, fighter cell, tries to fight with the invader, and they try to kill that or to get rid of it. But sometimes when our own body's immunity is low, it is not able to fight with that invader or the pathogen. So when it enters the body and it is very new for the body, like it was in this case of the coronavirus that affected the people in the recent time, it was totally new virus, and the people's antibodies that were produced in the body were not able to fight that new virus. And then what we had to develop a vaccine for that. Now, what is vaccine? Vaccine is a kind of biological preparation. So this is a kind of biological preparation in which we use dead and weakened microbes. So these dead and weak microbes, there is chemical substance which is prepared from these dead and weakened microbes, and then it is injected into our body, or sometimes it is given orally to the person. So what happens is when these dead and weakened microbes are introduced in the body, then the antibodies of the body try to recognize these dead and weakened microbes. They study those dead and weakened microbes. Because they are dead and weakened microbes, they are not able to produce or to develop any disease in our body, but it is injected into our body, introduced in our body, to make our antibiotics get prepared for the future attack. If in future any such microbe enter into the body, then our antibody will be able to recognize, oh, that is that same dead weakened microbe that was uh, in the past introduced in the body, and it will be able to fight against it. Okay, so that is the vaccine. And one important fact that you have to remember here, that first vaccine was developed by whom? This was developed by the Edward Jenner. He developed a vaccine for the smallpox. Edward Jenner was the first one to discover a vaccine, and he discovered the vaccine for which disease? He discovered the vaccine for smallpox disease, although uh, it is a disease which is now not no more there in the world. It has been eliminated from the world, and so this is the important fact that you need to remember here. So don't get confused between the antibodies and the antibiotics, but these terms are very important. Antibiotics, antibodies, and the vaccine, virus. I hope all this is clear to you. And but still there are many other interesting topics that are remaining in this chapter, like how the microorganisms contribute in the cleaning of the environment. And then there are various food preservation techniques which are important, and nitrogen cycle concept, nitrogen fixation concept is very important. This will be discussed in the next class. Thank you for watching the video. We will meet again with a new interesting topic, new interesting chapter.